Get started. Welcome to QAAFI's Science Seminar Series. I am the facilitator today. And before I start, Sandra, if you just pop it to the next slide. I'd just like to acknowledge the traditional owners and their custodianship of the lands that we meet today. And I pay my respect to their ancestors and their descendants who continue cultural and spiritual connections to country. And we recognise their valuable contributions to Australia and also to global society. My name is Heather Smythe. You would know me as being one of the sensory scientists and consumer scientists who works for QAAFI. I'm a researcher. I'm also currently the Deputy Director of Research for QAAFI. And I'm hosting, as I said, facilitating the session today. And I'm really happy to introduce to you Sandra Olarte. Oh, thank you, Sandra. Sandra Olarte Mantilla. Sandra has been working with me and with QAAFI now for close to 6 years. She has been a fantastic addition to certainly the group that I work with here, and she's situated with me out here at the Longpocket Elkhorn building. Sandra came to work with me after I'd had a really difficult 6 months, I'll be honest with you. It was a very dark time for me in QAAFI. I was absolutely stretched and had been working for about 9 months non-stops on delivering projects by panel, and Sandra was the amazing light at the end of the tunnel, and she came and in that time certainly our group has grown to have a number of postdocs and also a number of amazing technicians who work in sensory evaluation and flavour chemistry and consumer science. So I'm really pleased to have Sandra come and present to you today, and the title of her talk is The Journey of a Sensory Scientist and Consumer Scientist from an Agronomics perspective. Today, I'd like to just remind you all to please use the Q&A button at the bottom of the screen. The seminar will go from 12 until 1, but please don't put your questions in the chat, put them in the Q&A, and I'll be able to help Sandra walk through those at the end of her seminar. So please do encourage you to ask some questions. Thank you, Sandra. Over to you.
Okay, thanks, Heather. Sorry, can't see me yet. Yep, that's better. I too want to acknowledge the traditional owners and the custodianship of the lands on which we meet today and pay my respect to their ancestors and their descendants. So thank you for the opportunity to speak today. Yeah, as Heather said, we are based at the Longpocket campus at the Elkhorn building, and here you can see a picture of our beautiful building that, yeah, if you are able to come and visit us, we will be very happy to have you. And yeah, I named it, the title of my presentation as The Journey of a Sensory and Consumer Science, Agronomist in QAAFI, because, well, my degree is agronomy, and I believe that this combination has really helped me, like being agronomist and sensory and consumer scientist has helped me to understand the sensory trials better with that agronomy background. Just wanted to give a little short bio from myself. As I said, I have a degree of agronomy from the National University of Colombia. I did my honors in potato cultivars, Andean potato cultivars, a simulation of the model validation. And I did a few things before I moved to Australia, but I really was in love with wine science. I wanted to learn, and I moved to Australia. I did a diploma of horticulture, and I had the opportunity to work at Pfeiffer Wines for 2 years as a laboratory technician and cellar hand. And in that time, I met the supervisor of my PhD while I was judging wines in Rutherglen, Victoria. And this is what it led me to my PhD at the University of Adelaide. That I finished, I did conducted a project called Understanding the Relationship between Berry Sensory Assessment and Wine Quality in Shiraz. And yeah, after that, I was completely hooked on sensory. while I was doing my PhD, I did also a vintage at the Rainbow Vineyards in Tasmania. And well, I had the fortune in 2016, as Heather just mentioned, to start working with Heather as a sensory scientist. And it's been really fun, really different, busy. Yeah, and really, really grateful for all this time and all that I have learned from Heather. Amazingly grateful. So during these past 6 years at QAAFI, my role, I've been involved in doing so many quantitative and qualitative sensory and consumer studies, mainly on projects, linkage projects, HIE, Horticulture Innovation Australia, projects like the Natural Nutritious Project and Research Connections. And well, here you see an example. The linkage projects were mostly on snacks, beverages, and semi-solid foods, some model solutions, but mainly focus on texture of these products. In the Horticulture Innovation Australia, we work on biofortified horticultural products, and I did a consumer survey on perception of macadamias with high, low and high fat, and focus groups on the biofortified purple sweet corn. Also, we did studies on honey and other native products such as wattle seed and wattle seed bread that I will talk a little bit later more. And a big part of the focus of our group is understanding the influence on individual difference on texture perception and consumer preference. And as an example, how the number of taste buds that you can see here in this tank, how it influences texture perception. I will talk a bit more about that later in the presentation. So my role, my current role in the last 2.5 years is being divided into ARC projects, a linkage project, and the training center for uniquely Australian foods with Heather, Jasmina, and Jason Stock, which I'm really well grateful. And the role at my role at the ARC Training Center for Uniquely Australian Foods is being focused on different roles, helping the students, the PhD students in the sensory and consumer trials, also conducting some of my sensory and consumer experiments within the training center, and product development of some products, food products with native ingredients, and engagement with indigenous communities that I have really enjoyed, learning from them and teaching a little bit. One of the first projects that I conducted when I arrived in 2016 was this project with Be Inventive with honey. They have this beehive called the flow frame, which is beehive that the beauty of this beehive is that to harvest the honey, the only thing that you need to do is put this tab here, open the frame, and the honey will flow without having to disturb the bees. They don't have to come out. You don't have to get the frames out or anything. And well, Grace from Be Inventive, she was convinced that the honey was better, but she needed to prove scientifically that it was. And she wanted to know the sensory properties. So she brought her honey, and this is what the honey will look like from the yellow pea. She brought honey from 2 floral sources, yellow pea and macadamia, and also the honey from a semi-commercial beehive. And as you can see, it differs in color. So you kind of already could see that there will be differences. So what we did was to conduct a sensory profiling with 12 trained panelists. And because as you saw before, the honeys differ in color, so we have to do the assessment under red light to mask the color. And allowed the panelists just to focus on the aroma, flavor, and texture of the honeys and find differences. So what we found was that the honey from the flow frame was better. In terms of the macadamia honey had higher ratings, or from the flow frame had higher ratings of floral aroma, confectionery flavor, and lower ratings of resin dark than the semi-commercial honey. The yellow pea honey had higher ratings for cleanness and lower ratings for dry fruit mix pungency and lingering ratings than the semi-commercial. You can see in more detail the results of this study in the publication that I'm listing below. And one really good outcome from this study is that it led to be inventive to join the Uniquely Australian Foods Training Center for Uniquely Australian Foods. And we have a PhD that is conducting a PhD study that is conducting a project now. Another really fantastic project that we conducted that I was part of within my beginning of my experience with the with QAAFI is on water seeds. So we have these 4 species. As you can see, they look quite different. The size difference is real with the one that you see here, Cordiana and Coriacea. And we did a sensory profiling again with trained panelists, and we found that although the 4 of them have savory notes, they're described by the panelists with savory notes, they all have the different profiles. So for instance, Sophoria had a Vegemite note, Reti notes here had more like a peanut, Coliana more roast chicken, and the Coriacea mashed vegetables, coffee-like, fruity. So it was amazing to get these results and this difference. And we had a publication presenting these results and also the compositional attributes and nutritional values of these 4 species of water seeds. So this was done in conjunction with our industry client, Karen Sheldon Catering from Darwin. And also as a result of this project that was part also of Research Connections, Karen Sheldon has joined the training center. And we have a PhD student, 2 PhD students actually doing their PhDs with water seeds. One is Adiamo, and the other one is Sarah Jacobs. A second part of this study with Karen Sheldon and Research Connections was the development of a water seed bar baked bread that they wanted to be developed for a few reasons. And one main reason was to have an option of a more healthy bread for communities in Northern Territory and in Alice Springs, because they have a lot of trainees, indigenous trainees, and well, the options of bread is just like a white bread, which is not nutritious at all. So we did a few prototypes of the bread. The water seed that best was suited for the bread was the Coriacea, as you can see in the picture. And we did a consumer study with 100 consumers. And here you can see some pictures of how the breads were presented. We presented half bread to the consumers. And as a result, we found that the bread with water seed that was most liked by the consumers was with one of the highest concentrations of water seed. And also, we lately have been bringing this water seed bread to schools in Brisbane, and the kids just love it. And we are really surprised that this demographic, well, really love it. That's great. And one great benefit as well of this project is that the people who get to make the bread at Karen Sheldon in the kitchen and Parliament House are the trainees, employees, indigenous employees from Karen Sheldon, which is fantastic. And some of the customers are Qantas. So that bread is at the Qantas Lounge in Darwin. And we hope that Karen Shelburne is able to spread this product all over Qantas and other companies. As I said, also I'm supporting some PhD students in their studies. So with Sukita, we conducted a sensory experiment on allophytes. We selected 2 species, samphire and saltbush, from different regions, from different locations. And the idea with these allophytes is that they could be used as a salt replacement substitute. So I'm not going to be presenting any of the results here because Sukita is writing her paper right now. But yeah, just stay tuned for the results. And also for gaining, we conducted recently sensory profiling of Burdekin plum, which is a plum that is native to Australia, Queensland in particular. And we did the sensory profiling of this plant, and we developed a product. And we did the sensory profiling of this Burdekin plum product that I'm not allowed to talk about it just yet. But we are also preparing a consumer study of this product that hopefully you'll be able to taste soon. Another part of my role as in the training center is that, well, I have developed a couple of products in conjunction with indigenous owned and led enterprises with Australian native ingredients that unfortunately, because, well, we are just finishing and it's in confidential, I cannot say what they are, but they are soon to be launched and hopefully you can try them very soon. They are just delicious. And a part that I really enjoyed from being a part of the training center is the opportunity that I have to engage with indigenous communities. In 2019, we conducted a workshop with indigenous trainees from UQ. And it was really good to be able to show them a little bit of the science. So we conducted workshops with the sensory laboratory and also in the microbiology and the chemistry. And we also designed a lunch with native ingredients that they just love it. And it was really incredible that some of that they haven't even tried some of these native ingredients. And I just hope that, well, that changes because they are other products. And also, well, here you can see a picture of well, Kinari used to work with us. And we went to present the wattle seed bread to the indigenous communities and trainees that they go to Alice Springs to train the indigenous communities on native ingredients and the bread. And here you see also some pictures that when we went to Darwin last year in April with some indigenous stakeholders and funding bodies to exchange ideas and knowledge about native ingredients. And it was just wonderful to have the opportunity to see the land of, for instance, Michelle on here, showing us together with Bruno trees of kakadu plum and a green plum. And well, lastly here, it was really a pleasure to be able to show Josh and learn from her some of her experiences. And here just showing her a little bit of science in the lab. Another part that, well, is a particular interest of mine is edible insects. So in 2019-20, had the opportunity to conduct this project with a summer student, Chan Mugan and Lapagan. And we did a study together also with Lo and team of the Uniquely Australian Foods on the nutritional composition of green ants. And we found that the green ants differ in the composition in the different body parts, but also as a whole, they have high antioxidant, antimicrobial activity and folate content. Also with Daniel Cosolino, we used mid-infrared spectroscopy to discriminate the ants by the body part, and we are able to discriminate by the body parts. And also a volatile composition, we were able to discriminate between the different body parts of the green ants. And yeah, well, the publications, you can see more detail of this study in these publications. So yeah, as I mentioned before, I was part of a lot of linkage projects with Fonterra and PepsiCo. Unfortunately, many of them are still in preparation, the manuscripts. But basically what we did, we have beverage, snacks, semi-solid foods, evaluating texture perception. Texture is really important. It could be a really important driver of consumers. So that's why it's important to understand it. And so we did sensor profiling experiment and consumer experiments. And with a team of chemical engineering, they did on the same samples physical measurements such as rheology to evaluate the viscosity, tribology, friction. And we also, from these consumers and panelists, evaluate human individual difference, such as oral physiology, food behavior, like as was mentioned before, count the taste buds, the flow rate of the saliva, and whether they are food neophobics, if they like new foods or not. One publication that you can check some of this work is the one that I'm listing below, that we use an instrument that is called ring shear tester. And we found that the measurements of this equipment, of this technique, were correlated to sensory attributes such as sharpness and ease of clearance while during the mastication process from 4 potato chips, commercial potato chips that were manufactured differently and had different textures. So this is quite important because it could help the industry to design better food products depending on the needs of the market. Well, here there is a project that I am able to talk because it's been published, and is this work that we did with Fonterra in our ARC linkage project, and it's about particle perception in semi-solid food, and more particularly in yogurt. And well, first of all, why particle perception is important? It's important because even if you don't believe it, it's very common to have particles in food, whether they are occurring due to natural processing or they are added for different purposes. And yeah, the interesting thing as well is that we as humans, we are able to detect particle size in food, semi-solid foods, as low as 19 microns, and they can be detected in chocolate as gritty. So we've been studying particles within our group with trained panelists, and we found that the ability to detect those particles in semi-solid foods depends on different factors such as the particle size, the hardness of these particles, and the concentration and the matrix of in which those particles are added. So after this work with trained panelists and this knowledge, the logical questions and the logical flow of this work was to determine whether particle detectability will influence acceptability, and in this case, yogurt. So that is our question. Will it affect acceptability, particle detectability? And after that, the next question will be is whether individual factors are associated to consumers' ability to detect particles in semi-solid foods. To answer those two questions, we designed a consumer study with a systematic set of samples. So we have 9 samples. We have 9 yogurt samples. One sample was the control that we didn't add particles. And the particles were laboratory manufactured agar macrogels. As you can see here, this is how they looked. And we have, well, the particles had 2 levels. The set have 2 levels of hardness, whether 220 kilopascal or 550, a particle size between 30 and 100, or between 100 and 250 microns, and to levels of concentrations 2 or 5%. To make examples, so we add the agar macrogels to the yogurt, that in this case was the selected yogurt, a Greek-style yogurt from Rybeck Foods. And we selected this yogurt from an array of yogurts that we found at the supermarkets in Brisbane. And the particular thing with this yogurt is that it didn't present particles, it wasn't too sweet or too sour, and when we stirred to incorporate the particles, it didn't split. So we recruited via a survey 117 yogurt consumers, and we invited them to come to our sensory laboratories and to taste the non-yogurts and rate their acceptability in a scale from greatest imaginable dislike to greatest imaginable like. And a second task on this tasting was to taste the yogurts again and determine whether they could perceive the following 5 attributes: particles, drying, smooth, sweet, or no particles in each of these 9 yogurts. As a result, we found that, yes, definitely particle detectability influenced consumer acceptability. As you can see here in this plot, we have acceptability here, and is associated to the samples that have softer particles, and opposite to the samples that have harder particles with high 550 kilopascals, and associated to the attributes drying and particles. And the acceptability was higher and associated to the attributes sweet, smooth, and no particles. We also identified a detection threshold for particles around this sample set, that it was the sample set at about 50% of the consumers could detect particles, and also identify a rejection threshold for particles, which was the sample set at which the acceptability significantly declined. More details on this part for acceptability, you can find it in this publication. So for the second question, as I said, we wanted to know if individual human factors will be associated to consumers' acceptability to detect particles. So to do that, in the same 117 yogurt consumers, we invite them to come again to the sensory laboratory within a period of 2 months of the tasting. And we ran a series of collection of data, and they needed to complete some questionnaires as well. Some collection of oral physiology, and for example, we took pictures of the tongue and counted the fungiform papillae, which are these ones that are bigger and have like a shape of mushrooms. And in the first centimeter of the tongue and the second centimeter of the tongue, also we collected the saliva flow rate and stimulated saliva. And also we asked them to rate the intensity of prop, which is a beta compound, and a salt impregnated disc. And the important thing is, because in literature, it is a lot of literature mentions that individuals that rate are sensitive to the bitterness of prop are also better at tasting certain products and texture. Also, we collected mouth volume, and what we did was we asked the participants to we wanted to know how much they could hold, how much water they could hold in the mouth. So that was the mouth volume capacity. We also wanted to measure the tactile sensitivity and acuity. So tactile sensitivity, we measured it with a 2-point discrimination test, which is this disc that has 2 prongs at different distances and 1 prong. So what we did here is that we prong the tongue, and the participant was blinded. So we did two or 1 prong, And we asked the participant to tell us if they saw 1 or 2. And we determined their threshold. For the letter identification, we gave them some letters, different sizes that were embossed on some pieces of plastic, and they needed to identify with the tip of the tongue what was the letter. And we determined the threshold. Also, they fill some questionnaires, one for personality, whether they have an open personality, an extrovert, or more not so extrovert. Food choice questionnaire, whether their food choices are determined by sensory attributes or maybe something like environmental issues. Food phobia, whether they are open to try new foods or not so open. So there were different questions to determine their consumer behavior. And also we had questionnaires for food choice experience. So we have a questionnaire for the dairy products that include milk, cheese, and one particular one on yogurt products, how they consume their yogurts, where they buy the yogurts. Also a questionnaire that we created for yogurt texture preference and the textures that they preferred, whether it was runny or thick, for example. To analyze the data, what we did, based on the first part of the study with acceptability, we divided the consumers into groups. One group was the consumers that were able to detect particles or the attribute drying for each of the samples, and the ones who did not detect those two attributes. So we did divide them in those two groups from each of the 9 samples. And what we did was the statistical analysis, one way ANOVA, comparing, for instance, all the human factors, for instance, the number of taste buds for each of these 2 groups. So to determine if a human factor was considered to be influencer of particle detection, they have to be differ significantly in 2 or more samples in the same direction. So as a result, summarizing, we found that particle detectors significantly differ from non-detectors of particles by having higher fungiform papilla density and higher salt ratings. They also had higher oral tactile sensitivity by 2-point discrimination tests in the posterior part of the tongue, so in that section. So with this prong, they were more sensitive. They will have the distance was smaller for the ones that were able to detect particles in this area of the tongue. And also these particle detectors had an open type personality type. And they tended to have consumed the yogurt with cereal and smoothies. And the yogurt texture that they prefer were fatty, spoonable, and not separated. So more details on methodology and everything of this study, you can find it in this publication. But as a conclusion, I can say that, yeah, definitely human factors has an influence on particle detectability, but it's not just one factor. It's a combination of all these factors. Product experience, consumer behavior, and oral physiology traits and tactile sensitivity. Also, in our study, we found that, as I say, the 2-point discrimination test at the midpoint of the anterior tongue, the 2nd centimeter, is a key role in individuals' particle detection. And opposite to what the literature is suggested, particularly in terms of sensitivity to the bitter compound probe, saliva flow rate, and mouth volume and tactile acuity were not associated to particle detection. So yeah, why is this result are important? Why are these results so important? Well, these results are valuable to inform sensory and product development teams in the rational design of semisolid foods containing particles. For instance, if we find that if a particular market, a particular demographic tend to identify easier the particles, so you have to probably design your yogurt with smaller particles, softer particles, whereas another demographic doesn't mind, so they can design products for these markets different way. Also, it is important to know this, and in particular, we are preparing at the moment a manuscript evaluating the influence from an experiment that we did, evaluating the influence of human factors in 24 panelists in the performance in the discrimination of sensory attributes that will be published soon, hopefully. And this is important for us because our sensory panel at Elkhorn in our sensory team is a sensory panel that is in high demand in Australia. HEDA receive inquiries weekly. And so we really need to know our panel, the characteristics of our panel. And it not just for us, because it also is a panel that we share, a sensory panel that we share with DAF, with the Department of Agriculture and Fisheries sensory team at Cooper's Plains. So we both need to know our panel. So it's a really important asset. So yeah, that takes me to the end of my presentation. And I just want to acknowledge the sensory team at Elkhorn, Birndin, HEDA, Emma, Chaoyan, CNAFS staff, Jasmina, and Maral, chemical engineering, HEDA, Jason, and my industry partners that I was involved, Fonterra, PepsiCo, Be Inventive, Karen Sheldon, Andrew Cusack, and Maggie at DAF, the trade panel consumers, our funding bodies, IRC, Research Connections, and HIA. Well, thank you very much. And that's the end.
Thank you, Sandra. That's fantastic. Really lovely to see the very diverse number of projects that you've been working on with us for the last 6 years. That's quite incredible. I might kick off with just a technical question. I think people might not fully understand why open personality trait might have something to do with someone's particle, I guess, acceptability in a product. Could you tell us a little bit more about the different types of personalities that there are and why you think the open personality traits seem to relate to some of your data?
Well, I think, I guess an open personality will be more flexible to tolerate the particles. Even if you perceive it, I'll see it and I will tolerate it is what we will have strategies. For instance, we found that, well, this open personality type tend to have the yogurt with the muesli. Well, there is the particle, we'll find strategies. So whereas a personality type that is not open and is more cautious will not tolerate them. So so it's important to know not just the physiology of the consumers, but also the personality.
Absolutely.
For the industry to find strategies to please those other personality types. Yeah.
Correct. And also the fact that your personality type as a panelist will impact the way that you use scales. It'll impact the way that you that you interact in the study and your behaviour as a participant. So it's absolutely, I agree. It's quite logical that your personality type can impact the way that you score things. We have a question online here, Sandra. Thank you for a great presentation. You did your particle study on yogurt. How did you expect the viscosity of the food matrix to influence the detectability of particles?
Well, having done the experiment, and I would think that if the viscosity is if it's a less viscous matrix, let's say, I don't know, let's put an extreme matrix like water. Yeah. It will be easier to detect your particles, or maybe skim milk. You will detect easier the particles. Yeah,
absolutely. Whereas something more viscous like yogurt,
it will be harder.
Yeah. And on that particle detection, I mean, obviously it's not just about yogurts that you're interested in. A really important, I guess, product genre which is on the increase is and very trendy at the moment are plant protein food products. So things like meat analogues and also dairy or dairy analogues that use plant proteins, which are inherently problematic when it comes to particle perception. So how do you think your research might assist a field like that in the plant protein space?
Yeah, absolutely. I don't know if analogs, it could be sausages. That I guess is probably a matrix that is much more homogeneous. And if you're gonna try to simulate, I don't know, different components or replace, I guess you have to be really careful and really think about the perception of the different ingredients. What is the size that you're gonna be using in this replacement, these components?
Absolutely. Absolutely. Really important. Look, Sandra, I do have some of my own questions for you. I mean, I think it's so valuable to have an EMCR such as yourself with 6 years of experience in coffee. I suppose one of the questions I wanted to ask you was, in terms of your time in coffee, I guess, what did you enjoy most about your experience working with us and working with coffee in the last 6 years?
Well, I guess for me, the diversity of products and teams, the diversity in general, and the diversity of tasks. I definitely, there wasn't a moment that I was bored and I said, there was always anything new, everything, something different to do every day, like so many tasks. And I was excited, the different products that we try, beef, the model solutions, and the diversity for me was just really enjoyable.
Really exciting. I enjoy that aspect too, Sandra. It's always something different, isn't it?
Yes,
absolutely.
Absolutely.
So what do you think was your biggest challenge working with coffee on the flip side? What was tricky about working in coffee?
Well, I guess in my particular case, and due to the nature of my contracts, I guess, and well, it is what it is, and it's how it came. But that instability, like starting with very short contracts and not knowing, that part wasn't enjoyable, because I think that probably limited me a little bit. So definitely, that was the challenge. And I guess also being in different projects and with that short period contracts was hard also sometimes to deliver. And I suppose no excuses, but I guess maybe that makes it difficult to complete papers, I suppose, and grants and everything. So definitely.
Absolutely. So just for those of you online, Sandra's role was never, ever, not at any point 100% on one project. She was usually split across 3 projects at any point in time, but often it was 2 quite demanding different projects that Sandra was funded through, which certainly I can see challenges with that. And, the need to just deliver what you can deliver rather than always getting the time to spend to think about the fundamental research questions that you're trying to solve. You're trying to deliver to a partner, aren't you?
Yes, absolutely.
Quite challenging. So any other questions, pop them in the Q&A, please do. Otherwise, I'm going to keep quizzing Sandra with my little list of questions, which are a little bit selfish, perhaps, things I'd like to hear her talk about. So what, if you could turn back the clock 6 years to when you first started, Sandra, is something as an EMCR you might have done differently to the way that you, the last 6 years played out for you? Is there anything you would have changed or anything you would have focused more on or less on, anything you'd do differently?
Well, I guess if it wasn't at the same conditions of contract, I suppose we'll see now that I will do differently, but also try to get more support from QAAFI in terms of helping us to get more engagement with industry. Because, by being involved in so many projects with industry, I definitely understand that this is where we need to be at, and trying to write grants and generate grants. But trying to get myself engaged more with industry to get that funding, and I guess probably be more vocal with QAAFI management to definitely
have those opportunities,
help with that engagement. Absolutely.
So to build relationships with industry, you think more absolutely. Of that would have been key.
Absolutely. Absolutely.
Absolutely. Guys, any more questions? That'd be great. Pop them in the chat if you have any other questions for Sandra. Look, Sandra, I'll also ask you, perhaps this relates to my previous question, but what advice would you give for a EMCR starting out as a level A in coffee? What advice would you give them? And perhaps it relates to that, get industry connections might be your answer, but anything else you'd say to a new starter?
Yeah, I guess probably also the earlier that you realize, try to realize where you want to head your career. Yep. The earlier, focus on that. Not all of us need to be go the path of the professorship. Not all of us. So probably to start to realize that earlier. As well, if your path is within academia, and definitely start to build up those connections, or find the ways that help you to build those connections and with industry. Well, definitely look after your career, definitely, and definitely try to be involved in the activities that the university brings for your career, for develop your career. Yeah, definitely.
Definitely. That's something you've always been very good at, Sandra, is getting your hands dirty. You've been involved in so many communications exercises and events and whatnot, and hosting workshops and those types of things. You've really got into doing those. And also, you know, for many years now, you've co-hosted the QAAFI, the CNAFS DAF food group seminar series that we even share with CSRO now for many years, and serving on that has been, I guess, a great benefit to QAAFI, certainly to CNAFS, and I've certainly appreciated your efforts there to keep those relationships with DAF alive and help us to keep communicating with each other has been fantastic.
Yeah, absolutely. Yeah.
Any other questions in the chat? Oh, not in the chat, in the Q&A, guys, and I'm happy to ask Sandra. We've still got a few more minutes. Well, if there isn't any more questions coming through, Sandra, is there anything else you wanted to chat about?
No, I guess, well, I enjoy well, this experience, and well, definitely one part that I enjoy and that I learned from myself was that I could develop products, and I guess all that I learned from the different projects, not the specific objectives from the projects, but things around it helped me to develop the products, and that's something that I'm really happy. And
great.
Well,
that
find
you
that skill
didn't do before.
Exactly. Yeah, absolutely. Yeah. So that has been great, and as I said before, that engagement with the communities is an opportunity that not everyone gets, and that is fantastic.
No, absolutely. Yeah, well and truly, yes. Certainly, you weren't a product developer before you came along, but there's been a number of food products that you have developed, which are fantastic, and working at that interface with industry and to see those in market, it's really satisfying, and to see companies, use them in their business is certainly something that's we're all aiming for in coffee, aren't we? At the end of the day, we want these products to be consumed by humans and allow companies to get greater value out of their more agricultural products and ingredients. So that's been really good. Sandra, you've been fabulous to have in our team, and I just really lovely to see your work over so many years. You've done so many interesting things. You really have.
Thanks,
you're such a joy to have around and such a great team player. So everyone, thank you very much for attending today. I really appreciate it. And so does Sandra, I'm sure. The next seminar is if Sandra, if you would just move that one forward.
Now. Yep.
Is on the 21st of June. So please come along to that. Blood disease, an emerging threat to banana production, from Jane Ray, who's a PhD student with the Centre of Horticulture Science. So please come along to that if you can, everyone. Otherwise, thank you. I'll let you go 10 minutes early. I really appreciate you joining, and enjoy the rest of your day. Thanks.